Hello there, welcome to Biology 101. Today we will show you guys a simple experiment about photosynthesis and its evidence. Please enjoy.
To start off, let's give out a straightforward explanation of photosynthesis. Photosynthesis is a process performed by plants and some other organisms to make their own food. The reactants or ingredients for these processes are carbon dioxide and water with some help of light. The products would be glucose, a type of sugar, and oxygen. The chemical equation of this process is 6CO2 plus 6H2O will result in 6O2 plus C6H12O6, which is glucose. Light is not considered as a reactant because it's not a chemical substance, if not a form of energy. In other words, 6 molecules of carbon dioxide plus 6 molecules of water will result in 6 molecules of oxygen and a molecule of glucose. Now that we're clear about photosynthesis, we'll move back to the experiment.
The objective of our experiments is to demonstrate visually the process of photosynthesis by displaying one of its product, oxygen, highlighting the importance of photosynthesis in all our daily lives that we don't tend to notice.
The materials needed for this experiment are fresh spinach leaves, not necessarily spinach, if any fresh, green, broad, flat leaves from any plant, hole puncher, baking soda, in other words, sodium bicarbonate, liquid dishwashing detergent. You will only need a drop, so it's not that much, one plastic syringe, no needle, 10 milliliters or larger, 2 clear cups, 1 measuring cup, 1 measuring spoon, that is 1 1/8 teaspoon, and light source. Natural sunlight works, or you can just use artificial light.
So first of all, we used the measuring spoon to scoop out 1/8 teaspoon of baking soda and filled the measuring cup with 300 milliliters of water. Mixed the baking soda into the water, creating the bicarbonate solution. When the baking soda dissolved in the water, it released carbon dioxide. These would be used by the leaf for photosynthesis. Pour this solution into any cup and set aside. Next, we filled the clean measuring cup with 200 milliliters of water and added a drop of liquid dishwashing detergent to create a detergent solution. This solution would help breaking down the hydrophobic molecules of the surface of the leaf, therefore making it easier for the bicarbonate solution to penetrate the cells. Then we had to pour half of the bicarbonate solution into a clear cup. Place a drop of the detergent solution into the bicarbonate solution. If there were bubbles, it meant there wasn't enough bicarbonate solution. If not, it'd be perfect.
We then continue using the hole puncher to create 10 to 20 discs from the spinach leaf. Only punch holes in the smooth, flat area. We have to avoid large veins or the edge of the leaf. After that, we removed the plunger from the syringe and added the disc into the syringe.
After having the syringe prepared, we dip the tip of the syringe in a bicarbonate or detergent mixture and put in about 3 mm of liquid. Gently tap the syringe to suspend the leaf in the solution. Then we need to place a finger over the end of the syringe and pull back on the plunger to create a vacuum. And while keeping a finger over the tip to maintain the vacuum, we have to stir the leaf disc in the syringe for about 10 seconds and then eventually release the vacuum.
We had to repeat steps 8 and 9 to make sure the leaves took up the carbon dioxide. The disc sinking onto the bottom of the syringe indicated that everything was just fine. Finally, we poured the discs into the cup with the bicarbonate detergent mixture and watched them sink to the bottom. Lastly, we just needed to expose the cup to natural sunlight or artificial light just for several minutes.
As a result, the discs of the leaf would rise to the surface of the solution under the light. Because with light, carbon dioxide from baking soda, and water, photosynthesis was already taking place, which produced glucose and oxygen. Consequently, oxygen released from the discs of the leaf would eventually form oxygen bubbles that caused the discs to float. If anyone were to get rid of the source of the light, the discs would eventually sink because light, although not being a legitimate reactant, was vital for the photosynthesis process. An observation to be made was that since each of us used different leaves from different species of plants, some might create more oxygen bubbles while others less, because each plant had a different oxygen production portion and rate.
At the end, we'd like you to keep in mind of two ideas we want you to learn today. The first one, the reactants of photosynthesis are carbon dioxide and water, and the product are oxygen and glucose. The second idea is that glucose will be used as the fuel for plants later on. And on the other hand, oxygen molecules will be utilized during the photosynthetic processes, and some of them will be released to the surrounding environment.
As a conclusion, we hope you guys have learned that although the process of photosynthesis may seem unnoticeable, it is actually happening all around us, and that plants have an immense contribution to the air we breathe with the oxygen they give out.
As an extra exploration to our experiment, do any of you think that we might take advantage of the potential that the oxygen bubbles performed, carrying the disc to the surface as a form of energy or beneficial process? How? Maybe it can help in food factories' cleaning process when it comes to leafy vegetables. How about you? How can the oxygen bubble's ability be exploited?